Okay, students, good morning, all of you. Our next continuation topic, enzymes. Yesterday we discussed about enzymes. Enzymes. So, there are three basic tools are required. There are three basic tools are required. Among those, first one is enzymes, second one is cloning vectors, and third one is host organisms. Among those three, first of all, let us take discussion about enzymes. So, enzymes, restriction endonuclease, enzyme, which are two types. One is exonuclease, another one is endonuclease. Exonuclease, which can cut the DNA from the end of the DNA, end of DNA, from the end region of the DNA. From the end region of the DNA, exonuclease can cut. Endonuclease, which can cut middle of the DNA, middle of DNA. Exonuclease, endonucleases. Okay, so that is exonucleases and endonucleases. So generally, restriction endonuclease enzyme can cut the plasmid DNA as well as foreign DNA, desired DNA, or alien DNA, etc. So already yesterday we have seen that. Like this, restriction endonuclease enzymes can cut. Restriction endonuclease. So like this, endonuclease can cut within the DNA. That means middle of the DNA at the specific locations. Whereas it could not cut at the specific locations of the plasmid DNA as well as foreign DNA can cut. So when restriction endonuclease enzymes can cut plasmid, it leaves single-stranded portions with complementary. Single-stranded portions with complementary. That complementary T, G, G, any complementary. It does not exactly this is only A, C, C. Any complementary would be there. This is plasmid DNA vector. Next, foreign DNA. Foreign DNA can cut this. Okay. T, A, G, C, G, C. A, T, C, G, C, G. This is called foreign DNA, alien DNA, or gene of our interest. Which gene, the plant or animal, which has a superior characters, from the superior character plant or animal will take DNA, that is foreign DNA or a desired DNA or gene of our interest. So in both the cases, restriction endonuclease enzyme can get the DNA according to the complementary base pairing. Restriction endonuclease enzyme can cut plasmid DNA as well as foreign DNA at a specific sequence of DNA, specific nucleotide bases according to the complementary. So that after cleave, in both the cases, a single strand is called sticky ends. Sticky ends. This is also sticky ends. So that they can leave single-stranded portion, single chain. That is called sticky ends. This is sticky end, this is sticky end. Both are complementary with each other. That is the first restriction endonucleases. Restriction endonucleases enzyme. Next, DNA ligase. It is also called sealing enzyme or joining enzyme. Sealing or joining enzyme. Sealing or joining enzyme. That is called DNA ligase. Sealing or joining enzyme. Sealing or joining enzyme. See, as a result of restriction endonuclease cleave the foreign DNA as well as plasmid DNA. As a result of cleavage of plasmid DNA as well as foreign DNA with the help of restriction endonucleases. Those cut fragments, those cut fragments combined. This is who will be combined here. This is who will combine with this. As a result of cleavage of plasmid DNA as well as foreign DNA, who will make together, who will bind together, who will unite together with the help of enzyme that is called DNA ligase, sealing enzyme, binding enzyme, or joining enzyme. Object to discussion bit. Okay, so DNA ligase is a binding enzyme or joining enzyme. Okay, that's all. So that is the DNA ligase enzyme. So then, see here, let me show this is the picture. So this is okay like this way. So this part we will bring and we will combine. So if we will combine, how it will be? See that a single circular like this way. Okay, like this way. Single circular. It is a single circular plasmid is formed. This plasmid contains its own DNA as well as foreign DNA. This is called recombinant DNA. As a result of binding or sealing the plasmid, single circular autonomously replicating plasmid is formed. Single one circular autonomously means they themselves replicate. Autonomously replicating plasmid is formed. Therefore, it is called recombinant DNA. The word itself saying the significance. Recombinant DNA, recombined DNA, recombinant DNA. See, recombined means from another organism we brought DNA. The DNA is a superior DNA. That superior DNA we are bringing and making attached with each other. So that is called DNA ligase. Even this DNA ligase enzyme also collected from bacteria, different types of bacteria. This is that. Next one is DNA polymerase. It is also called synthesizing enzyme. It is also called synthesizing enzyme because object to discussion. DNA polymerase nature is what? It undergoes replication of this DNA. Both will be integrated. Okay, both will be integrated, undergoes replication with the help of DNA polymerase and produces number of copies. Those copies are called clones. Okay, in that one, from the existing DNA, so in this one, plasmid DNA, foreign DNA, these two togetherly combine and replicate with each other and produces new strands of DNA from existing DNA. New strands are forming through replication. New strands are formed through replication. Therefore, it is also called synthesizing enzyme. Okay, so even DNA polymerase enzyme also collected from the different kinds of bacteria. That's all. So that is the DNA polymerase. DNA polymerase enzyme. DNA polymerase enzyme. DNA polymerase enzyme, it undergoes, it induces existing DNA to form new DNA through replication. Therefore, number of copies of new DNA, multiple copies of desired DNA are formed. Those desired DNA contains antibiotic resistance. That means resistance against antibiotics. That is called DNA polymerase. That is why it is also called synthesizing enzyme. Lysing enzymes. Lysing enzymes. The word itself says the meaning lysing, lysis, nothing but split. There are some enzymes can cut the cell wall to get the DNA. Those enzymes are called lysing enzymes. There are different types of lysing enzymes. That is lysozyme, cellulose, kinase, kinase. These are called lysing enzymes. There are certain enzymes acts as a lysis. Okay, these enzymes break the cell wall, cut the cell wall, or open the cell wall to get DNA. Lysozyme, it is used to break the bacterial cell to get the DNA. Lysozyme, break the bacterial cell. Break the bacterial cell. Cellulase. See my is by mistake I wrote cellulose, not cellulase enzyme. Cellulase enzyme. Cellulase enzyme. Cellulase enzyme can break the plant cell wall. Can break the plant cell wall and we can get DNA. Kinase, it is the enzyme which is used to cut the fungal cell wall to get the isolated DNA, separated DNA, to get the separated DNA. Okay, so these are called lysing enzymes. Lysing enzymes. Next one is alkaline phosphatase enzyme, which is used to break the DNA at 5' end region, 5' end region, which is used to break the DNA from 5' end region. 5' end region, if you wanted to cut the DNA, they will use alkaline phosphatases. Like this way, different kinds of enzymes are used to break the cells and isolate the DNA. About the isolation of DNA, there is a separation topic is there. Separation and isolation of DNA. So where is the DNA is not available alone. DNA is always associated with macromolecules, histone proteins, non-histone proteins, polysaccharides, RNA. With them, it is mixed. One by one proteins, non-histone proteins, polysaccharides, macromolecules, one by one, we have to remove, and finally, alone, we have to isolate a pure form of DNA. That is separate topic is there, so we discuss next chapter, next topic. Okay, so these are all called enzymes. Okay, so among the tools, first tool is enzymes. Those many of enzymes are needed. So in your textbook, at one particular place they did not do. So what I'm saying, when enzymes matter has come about that, all the enzymes and activate. These all the enzymes are involved in biotechnology. Okay. Next topic is, this is first two, second two. Cloning vector. Cloning vector. It refers to the cloning vector or gene carrier in which desired gene has been inserted. Cloning vector means what? Plasmid. 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 Cloning vector, plasmid. Cloning vector, plasmid. Okay, see, plasmid. It acts as a carrier or vector or gene carrier. This is plasmid. Yes, that's okay. So it is complementary. Plasmid. It is acts as a vector. Cut plasmid. It acts as a vector or gene carrier in which foreign DNA has been inserted. 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 So that autonomously replicating recombinant DNA are formed. It undergoes rapidly multiplying or amplified and producing multiple copies of desired DNA. Multiple copies of desired DNA. Those desired DNA we collected and inserted into less superior plants or animals. Then those plants are called genetically modified plants. Genetically modified plants or genetically modified animals. Those plants will be transgenic plants or transgenic animals. So there are a number of cloning vectors are number of. Cloning vectors are number of. Those are first one is plasmid, already I wrote. Plasmid. Plasmid is an autonomously replicating circular extra chromosomal DNA which is present in bacteria. Salmonella, Typhimurium bacteria. Not only Salmonella, Typhimurium bacteria. Any type of bacteria plasmid will be there. The plasmid will be used in genetic engineering experiments. Okay. Next one, second cloning vector is bacteriophages. 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 Bacteriophages means what? Some of the viruses that infect bacteria. So such type of viruses are called bacteriophages. Third one is Artificial chromosomes. Artificial chromosomes of bacteria, yeast, etc. Artificial chromosomes of bacteria, yeast, etc. etc. means here and mammals. Some of the mammals also used as cloning vectors. Artificial chromosomes of bacteria or yeast or mammals. Chromosomes. Exactly related to that chromosomes. Artificially they will synthesize and used as vectors. You may be surprised. So some of the vectors scientists are artificially synthesized in the laboratories. That's why artificial chromosomes of bacteria, yeast and mammals. Next. Transposons or jumping gene or mobile genes. Jumping genes or mobile genes. So why we are calling transposons? Transposons also called jumping genes. Jumping genes also called mobile genes because DNA sequence, which is often it changes its location in some of the microorganisms. Okay, DNA sequence, which is often changes its location in genes of some microorganisms. In such organisms, genes are called transposons, jumping genes, or mobile genes. Mobile genes. Okay, these are all acts as cloning vectors. Among these, very famously we use plasmid and plant animal viruses. Plant and animal viruses. Of the above mentioned cloning vectors, the most famously plasmids and animal and plant viruses are most extensively used in genetic engineering experiments. Genetic engineering experiments. Okay, that's all. So that is the topic. Okay. So this is the enzymes. Next one is the next tool, cloning vectors. Here, 5 marks question they will ask. Briefly explain about the tools of recombinant DNA technology. At that time, first you have to mention about enzymes. Next, cloning vectors. After that, now I am going to say that. Okay. Third one is third tool. Host organism. So for recombinant DNA technology, it is recombinant DNA. For recombinant DNA technology, soon after identifying plasmid, and formation of recombinant DNA, the foremost step, the major step is to transfer this recombinant DNA into the host cell or organism. Host cell or organism. Host cell or organism. Okay, this is the basic step. Host organism. Yesterday I said that. So when recombinant DNA is formed, it's needed to be transferred. Escherichia coli bacteria. So this is entirely transferred into the Escherichia coli bacteria. Why? As Escherichia coli reproduced. What reproduced? Same copies of desired DNA by replication. Okay, as E. coli reproduce and it can be formed clone of cells. Clone of cells. Clone of cells means what? Same copies. Number of copies are formed. So that this recombinant DNA is needed to be transferred into the host cell or host organism to multiply or amplify desired DNA fragments. That is, so after replication, it undergoes replication with the help of DNA polymerase and produces a number of copies of desired DNA fragments. So then, when it is transferred into the E. coli bacteria, it would not be visible. It would not appear. So this is plasmid DNA and foreign DNA are integrated and produces number of multiple copies. The process of production of multiple copies of desired DNA fragment is called cloning. So number of DNA fragments are formed. Number of DNA fragments are formed. Millions of copies. 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 Like this, millions of copies are formed. Okay, so that's this is in recombinant DNA technology. So now after identifying plasmid DNA and formation of construction of recombinant DNA, the major step is to transfer this recombinant DNA to the host cell or organism for the multiple DNA. So this can be employed with the help of two methods. That is, one is direct method, another one is indirect method. Direct method and indirect method. So transfer of recombinant DNA into the bacteria can be done by two methods. One is without using any, manually we cannot send. Directly they will send one method. Indirectly through some other equipments they will send. That is we will discuss later. Later we will discuss. Once again, this topic we needed to keep discussion. At that moment, direct method. How recombinant DNA is transferred into the E. coli bacteria. That is what at that moment we will keep discussion. Okay? Okay, next. Restriction endonuclease. Restriction endonuclease enzyme. Okay, in this one, in genetic engineering experiments, the most important step. In genetic engineering experiments, the most important step is to cut the plasmid DNA as well as foreign DNA. That is the foremost step. After killing the plasmid DNA and foreign DNA, the next step is incorporation of foreign DNA permanently into the plasmid DNA, so that autonomously replicating circular extra chromosomal DNA is formed. Okay, that's all. Every moment. So whatever may be the diagram I am drawing, so the same diagram when we will finish this chapter, nearly many times I need to be drawn. Many times I need to be drawn. Okay, that's all. Okay, many times like this way. In genetic engineering experiments, the foremost step is to identify the plasmid DNA. After identification, cut the plasmid DNA as well as foreign DNA. so that foreign DNA can be incorporated permanently into the plasmid DNA and forms autonomously replicating circular extra chromosomal DNA is formed. Okay, so after insertion, disease becomes circular. It becomes circular. Okay, so now it is called one circle. This is called one circle. One circle. Like that it forms. This is recombinant DNA. Okay, here in genetic engineering experiments, identification of DNA is very important. First, restriction in a nucleus engine can identify the length of the DNA. First, it will identify what is the length, how much its length. First, it will identify the length. Identify the length and cleave the DNA. Identify the length. Identify the length and cleave. Cleave the DNA at specific locations. Cleave the DNA at specific locations. Listen carefully. Cleave the DNA at specific locations. That cleaving process now I will show. For example, so this is a plasmid DNA. This DNA in the form of double helical structure we will show. This is plasmid DNA. Restriction in the nucleus can identify the length and cleave at the specific locations of the DNA. How it will cleave? Exactly. This part will cleave. Cleave. How it will cleave? A, P, C, G. It will cleave. Like that it will cleave. Now A, P, C, G. Cut. Like that. That cut part will be decided. This is a plasmid DNA. This is called sticky ends. Single stranded portion, sticky ends. Single stranded portion, sticky ends. Next, foreign DNA. Foreign DNA. GATTC. GATTC. This is desired DNA. So plasmid DNA, restriction endonuclease DNA can give plasmid DNA as well as foreign DNA or desired DNA at specific locations. At specific locations with the specific sequence. Plasmid DNA can cut AATCG region. Foreign DNA can cut GCPPA region. G, C, P, P, A region can cut. The cut portion I am showing. G. Like this. like this way. It is a single strand. It is a single strand. So this is how I have shown like this way. Without sequence, that could be appear like this way. In DNA sequence, like this way it appears. Okay. So after cleave, so plasmid DNA and foreign DNA both were we cleave. Then what? With the help of DNA ligase, we have to combine. Then this is, we will take it and combine. Then it will come here, it will come here and combine. That combination I will show. A, P, P, C, G. Okay? Then now that I can modify it like this. This is called recombinant DNA. This is called recombinant DNA. Okay, restriction endonuclease. Okay, first restriction endonuclease enzyme can inspect the length of the DNA, identify the length of the DNA. After identification, it can cleave the DNA at a specific location with a specific nucleotide basis, that is G, A, T, T, C. It is complementary with each other. G, A, T, T, C. C, G, C, A, T, A, T, T, A, T, A, C, G. This kind of a complementary is called palindromic complementary. Palindromic complementary. Palindromic complementary. Palindromic means what? In English, palindromic words will be there. From left to right, right to left, same words are repeated. G, A, T, C. From left to right. From right to left, G, A, T, C. This kind of words are called palindromic sequence. Exactly according to the palindromic sequence, restriction endonuclease enzyme can cleave. It can cleave foreign DNA, plasmid DNA. This kind of cleavage is called palindromic sequence. For example, Malayalam. Malayalam. Same. From back to front, front to back. Left to right, right to left. Backward to forward, forward to backward. Same. Madam. Madam. Gadag. 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 This is called palindromic sequence. Palindromic sequence. Okay, like this, there are number of palindromic words are there in English department. Okay, like this sequence exactly cut by the restriction endonuclease enzyme. Restriction endonuclease enzyme. Today our meeting session is finished. So next continuation topic we will see tomorrow. You can leave the class.